Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay,
Okay.
a very good
A
evening to
very
you
good
all.
evening to you all. Good evening,
Good
sir.
evening, sir.
Good evening, sir.
Today we are
Good
going to
evening, sir.
today we are going to
We are going to
discuss
discuss
Mendel's
Mendel's
laws.
laws.
Did I share the PPT with
PT
you?
with you.
Just a
Just a
second.
second.
Please
Please
mute it.
mute it.
Okay, let's
Let's
start.
start.
So
So
today we
today
are
we are
going to
going to
discuss
discuss
about
about
the
the
laws
laws
proposed
proposed
by
by
or laws
laws
of
of
Mendel,
Mendel,
Gregor
Gregor
John
John
Mendel, who
Mendel,
is
who is
called
called
as
as
father
father
of
of
genetics.
genetics.
An
An
extract from a
extract from a
publication
publication
reads
reads
like
like
this.
this.
Pre-Mendelian
Pre-Mendelian
biologists
biologists
had
had
some
some
data on
data on
heredity,
heredity,
but it
but it
was
was
poorly
poorly
organized,
organized,
inadequately
inadequately
recorded,
recorded,
and
and
misunderstood.
misunderstood.
That is the
That is the
reason
reason
why
why
any of
any
the
of the
scientists
scientists
before
before
Mendel
Mendel
could
could
not
not
succeed
succeed
in
in
formulating the
formulating the
principles
principles
of
of
inheritance
inheritance
because
because
their
their
research is
research is
poorly
poorly
organized,
organized,
the
the
record is
record
said to
is
be
said to be
inadequate,
inadequate,
and
and
they are
they
said
are
to
said to
be
be
considering
considering
all
all
characters at a
characters at a
time,
time,
hence the
hence the
results were
results were
misunderstood by
misunderstood
them.
by them.
Whereas
Whereas
Mendel's
Mendel's
formulation
formulation
possessed
possessed
brilliance,
brilliance,
clarity,
clarity,
and
and
breadth of a
breadth of a
completely
completely
new
new
principle,
principle,
which gave
which gave
rise to
rise
the
to the
evolution of a
evolution
new
of a new
discipline, that
discipline,
is
that is
the
the
branch of
branch of
science
science
called as
called as
genetics.
genetics,
The
the
foundation of the
foundation of the
science
science
named by
named by
Benson
Benson
in
in
1906
1906
as
as
genetics.
genetics.
Okay,
Okay,
so
so
this is
this is
how
how
the
the
scientific
scientific
community
community
feels
feels
about
about
Gregor,
Gregor,
the
the
great
great
Gregor
Gregor
John
John
Mendel.
Mendel.
And
And
we are
we are
going to
going to
learn
learn
about
about
processing of
processing of
hybridization,
hybridization,
inheritance of
inheritance of
characters,
characters,
process of
process of
segregation,
segregation,
and
and
critical
critical
review of
review of
Mendel's
Mendel's
laws.
laws.
For
For
this,
this,
we have
we have
yesterday
yesterday
discussed
discussed
about
about
all these
all these
points
points
about
about
the
the
difference
difference
between
between
character and
character and
trait,
trait,
the
the
seven pairs
seven
of
pairs of
contrasting
contrasting
characters of
characters of
Mendel,
Mendel,
and how
and how
Mendel
Mendel
selected pure
selected pure
lines
lines
and
and
different
different
crossing
crossing
experiments like
experiments like
monohybrid
monohybrid
cross,
cross,
dihybrid
dihybrid
cross,
cross,
reciprocal
reciprocal
cross,
cross,
test
test
cross, and
cross,
back
and back
cross.
cross.
And
And
these are the
these are the
seven pairs
seven
of
pairs of
contrasting
contrasting
characters.
characters.
As we
As we
know,
know,
stem
stem
length
length
is
is
represented
represented
by
by
tall and
tall and
dwarf,
dwarf,
cotyledon
cotyledon
color,
color,
yellow
yellow
dominant,
dominant,
green
green
recessive,
recessive,
seed
seed
shape,
shape,
round
round
dominant,
dominant,
wrinkled
wrinkled
recessive,
recessive,
flower
flower
color,
color,
purple
purple
dominant,
dominant,
and
and
white
white
recessive,
recessive,
inflorescence,
inflorescence,
axial
axial
dominant,
dominant,
terminal
terminal
recessive,
recessive,
pod
pod
shape,
shape,
inflated
inflated
dominant,
dominant,
constricted
constricted
recessive,
recessive,
and
and
finally
finally
pod
pod
color,
color,
green color
green
is
color is
dominant,
dominant,
and
and
white
white
yellow
yellow
color is
color
said
is
to
said
be
to be
recessive.
recessive.
So these
So
are the
these are the
seven
seven
pairs of
pairs of
contrasting
contrasting
characters
characters
on
on
which
which
Mendel
Mendel
worked
worked
on
on
each and
each and
every
every
character.
character.
He has performed monohybrid cross for each and every character. He has even performed dihybrid cross for by considering two different pairs of contrasting characters. Okay. And Mendel selected for his experiment only pure lines because they are true breeding. We have to select only true breeding pure lines for the hybridization experiments. And this is possible in Pisum sativum because it is a bisexual plant, and self-pollination leads to the production of pure tall plant or pure dwarf plant or any other contrasting character. And coming to the laws of Mendel, there are two laws of Mendel. And in some books, it is mentioned that there are three laws of Mendel. One is law of dominance, the other is law of segregation, and the third one is law of independent assortment. These are the three laws mentioned in many of the books. But the scientists say that there are only two laws of Mendel, that is the law of segregation and the law of independent assortment. So whenever someone asks you how many laws of Mendel are there, you have to say the Mendel's laws are two in number. One is the law of segregation. This is called as the first law of Mendel. And the other one is the law of independent assortment, which is the second law of Mendel. And the law of segregation is also called as law of purity of gametes. If you don't know, you can write it down. Law of segregation is also called as law of purity of gametes. Okay, and law of dominance, which we generally have studied, is part of law of segregation. Okay, and now we are going to discuss about the law of segregation, which is based on monohybrid cross. Monohybrid cross, we know it is the cross between two plants differing in a pair of contrasting traits. For example, tall or dwarf, round or wrinkle, purple or white, axial or terminal of a single character. Like for tall and dwarf, the single character is stem length. For round and wrinkle, the single character is seed shape. For purple or white, the single character is flower color. For axial terminal, the single character is flower position. So every character in Pisum sativum are represented by a pair of contrasting traits, and these individuals, the plants differing in these two contrasting traits, were crossed in monohybrid cross by Mendel. Okay, and coming to the Mendel's experiment. So we have discussed about the Mendel's experiment yesterday. A pure tall plant, which is a pure line, is crossed with a pure dwarf plant. And in the F1 generation, he could get all the 100% tall plants only. Based on this, Mendel said that the tall is said to be appeared in the F1 generation because it suppressed the dwarf character. Okay? So based on this first experiment, first hybridization between a tall plant, pure line, pure tall plant and a pure dwarf plant, as he got only tall plants in the F1 generation, Mendel suggested that or concluded that the tall appeared because the dwarf was suppressed. And these F1 plants, which are said to be tall, are now selfed. Because it is a bisexual plant, it can be self-pollinated. So selfing is done. And all first generation tall are said to be hybrids. When they are selfed, in the next generation, Mendel got 2/3 of second generation tall are said to be hybrids. Okay? And the first of this 1/4 are said to be pure tall, and 1/4 are said to be pure dwarf, and the remaining 2/4, with respect to tall, There are three only, so two-thirds. Two-thirds of second generation tall are found to be hybrid by Mendel. Okay, so now we are going to discuss about Mendel's laws. Actually, what the scientific community says is that Mendel did not propose any law. Mendel explained the process of inheritance based on his monohybrid cross and dihybrid cross. And monohybrid, based on these two different types of crosses, he explained us the process of inheritance. He made some conclusions or deductions based on which he made four postulates. Mendel made four postulates. We can call them as principles also, of which the first three postulates are based on monohybrid cross, and the fourth one is based on dihybrid cross. Okay, so thus Mendel made four postulates only. But the scientists say that in 1865 to 66, Mendel proposed his principles of inheritance in the annual proceedings of the natural history present in Austria, which is maintained by the Society of Brunn. Okay, but because this is a journal or proceedings which is not found anywhere in the world, his discoveries are in dark till they were rediscovered in 1900 again. There is a small, brief, pathetic story of Mendel which I would like to mention here. What actually happens here, whenever you want to do a research, okay, whenever you want to do a research, what you have to do is you have to see whether that particular research is done by any other person in the world or not. If you don't check and go on with your research and you get some results and you publish, later someone points out that this research already has been done by some other scientist in China or America or Japan, Korea, England, way before 19th century itself, then what does it mean? Your research is a copy. So what every PhD joining student first they have to do is they have to check the publications for the past 50 to 70 years. For my PhD, I have checked for the past 70 years. And some scientists, some researchers go before 30 years or 50 years. Okay, and after that only I got confirmed that the topic that I have chosen for my PhD or research is not dealt by any other scientist in the world. Now I can boldly take the research topic and complete my research. Okay, that is what we have to do. Mendel has completed his research and he has submitted the research findings to the then scientific community, and most importantly to one Anton von Kramer. Okay, he is the one who coined the term protoplasm and cytoplasm. So he submitted his results to Anton von Kramer, and Anton von Kramer said that, okay Mendel, you got a good result. Why don't you prove this on another plant? Because you have done only this on one plant and you have proposed certain principles. We cannot show these results as universally applicable because you have done only on one plant. So why don't you go for another, the same research on another plant? So Mendel agreed to that and asked Kramer, why don't you suggest the plant itself? So he suggested a plant called as Heratium, okay, which is commonly called as hawkweed. It's a weed It's commonly called as hawkweed, Heratium. Mendel took the plant, went to the monastery, and started, already has completed 9 years of research, and now he has worked on this hawkweed for another 4 years. And to his astonishment, he did not get any result at all. The results that were got on Pisum sativum were not proved in hawkweed. And Mendel got very much disappointed, and in due course of time, he became blind and he died soon. Okay? And his research that was published in a small proceedings, annual letter we can call this, of a small society present in the Austrian country, which is not published anywhere else in the world. Because of this reason, his research went into the darkness until it was rediscovered by three scientists. De Vries, Correns, and Schermark. De Vries from Holland, Correns from Germany, and Schermark from the same place where Mendel has completed his research, that is Austria. So they have done this experiments on different types of plants and animals, and they have got the same results as Mendel did. So what actually do is, whenever you take up research, you have to check whether someone has done it or not. But these three scientists, though they have searched for it, they could not get anything because it was not a journal or publication that is distributed worldwide. They could not come across. So, but at last, after the entire research is over, at some point of time, maybe Schermark, who belongs to, this is my interpretation, who belongs to the same area, Austria, might have found this publication, and he shown it to all the others, and all the three scientists, De Vries, Correns, Schermark, came to a common conclusion that because a great man like Mendel has done this before 35 years that we have come up with our publication, we have to give full credit to the Mendel only. So they have given full credit of the principles of inheritance to Gregor John Mendel only. And out of these three, Mr. Correns, scientists say, Mr. Correns proposed two laws, that is law of segregation and law of independent assortment. For our convenience and for our understanding, the law of segregation is divided into law of dominance and law of segregation in later times. Okay, so this is the brief history of rediscovery of Mendel's laws. Are you following?
Yes, sir.
Okay, good. So now Mendel has taken what? Has represented this in the form of factors, isn't it? So let's see the first deduction of Mendel based on the monohybrid cross. What is his first deduction? Mendel first, what he has done? He has selected pure lines in parental generation. That means this tall plant is not a hybrid and dwarf plant is also not a hybrid. They are pure for that particular character. Tall can give rise upon selfing only to tall, and dwarf can give rise upon selfing only to dwarf. And all F1 plants are said to be found to be tall only. So we can say that tall is a dominant character because it suppresses the dwarf character, and dwarf is the character which is suppressed by the tall character, and he considered this as a recessive character. And this, based on this, Mendel concluded that out of a pair of contrasting traits, that is tall and dwarf, of a single character, that is stem length, only one appears in the F1 generation. The one which appears in the F1 generation is called as dominant character, and the one which is suppressed in the F1 generation is called as recessive character, and the factor responsible for dominant is called as dominant factor capital T, and the factor responsible for recessive character is called as recessive factor small t. And this is called as the principle of dominance. So, as I have said before, based on monohybrid cross, Mendel has made three deductions or three postulations. And first and foremost of them is the principle of dominance, which we call it as the law of dominance later. Okay? And coming to the next part of his experiment, where he has self-fed the F1 plants and got the tall and dwarf in 3:1 ratio, and he continued this to the next generation also. Let's see up to the F2 generation only what has Mendel made. So after continuing his experiment till second generation up to F2, based on these results, Mendel made second deduction. The F1 tall plants were self-pollinated to know why dwarf character did not appear in the F1 generation. Why the dwarf character that is present in the parents is not appearing in the F1? That might have been the question that have arose in the mind of Mendel. And to know that, he self-pollinated the F1 plants, and to his astonishment, he found dwarf plants in the F2 generation. These are the F2 generation dwarf plants. And based on this, he might have concluded that unless being present in the F1 generation, dwarf character would not appear in the F2 generation. Okay? So, it is passed on from the F1 individual to the next generation, F2 individual. That means the factor responsible for dwarfness, that is small t, might have been present in the F1 generation. And the F1 individual is not a pure tall. It is found to be, according to him, it is a hybrid tall. Okay? As F1 is hybrid, it carries two factors. One for tallness, that is capital T, and the other for dwarfness, that is small t. Okay? And based on this, he concluded that his second postulate or principle, that is the principle of unit factors. So what does the principle of unit factors say? For every character, what is this character here? Stem length. It is represented by a contrasting traits. What are they? Tall and dwarf. Because the hybrid is able to transfer or transmit the dwarf character to the F2 generation individuals, Mendel thought that this factor that is responsible for dwarfness might have been also present here in the F1 hybrid. Because tall also appeared, the factor for tallness is also present. Thus, for every character, there are two factors which determine it. And these two factors, we can call them as one pair of factors. So this one pair of factors responsible for a pair of contrasting traits of a single character are called as unit factors by Mendel, which were later called by us as Johansson said, genes. Okay? So what is the principle of unit factors? It states that there are a pair of factors responsible for the determination of a pair of contrasting traits of a single character. That is what principle of unit factors is. Okay? Next. Mendel continued his experiments till third generation. As we have discussed yesterday, why he continued his experiments till third generation? Because he wanted to know whether the tall plants in the F2 generation all are pure or any hybrid is present. So he continued his experiments till third generation, where the F2 four different types of phenotypes are said to be self-pollinated again. Upon self-pollination, 1/4 of the total plants or 1/3 of the tall plants gave rise to only tall. Based on this, we can say that the F2 plant, this F2 plant is a pure tall. And 1/4 of the dwarf F2 plants gave rise to only dwarf. We can say that this is a pure dwarf. But the remaining 2/3 of tall plants or 2/4 of whole F2 plants gave rise to tall and dwarf in 3:1 ratio. That means these two are hybrids. So based on this experiment where the hybridization process is continued till third generation, Mendel concluded that the second generation individuals here, some are said to be pure tall, some are hybrid tall, and the others are pure dwarf. And based on this, the genotype of the F2 plants is known. Now we call the pure tall plants as homozygous tall, hybrid tall as heterozygous tall, and pure dwarf as homozygous dwarf. Okay? So 1/4 of the tall plants, 2/4 are tall and dwarf in 3:1 ratio they gave rise to, and 1/4 gave rise to dwarf plant. Mendel called them respectively as pure tall, hybrid tall, and pure dwarf plants. And the genotypic ratio is 1:2:1. Okay? So let's see how it happened. The F1 tall, which is hybrid, F1 tall, which is hybrid, carries two factors, capital T, small t, isn't it? Capital T and small t. So the F1 is self-pollinated. This part is the androecium of the F1 plant, and this part is the gynoecium of the F1 plant. So both have capital T, small t genotype only. And now what happens? During the formation of gametes, what happens here? One of the two factors enters one gamete and the other factor enters the other gamete. Likewise, the same thing happens here. Capital T enters one gamete, small t enters another gamete. So these are the pollen grains and these are the ova. During formation of gametes, both dominant factor and recessive factor separate from each other. They separate from each other and they enter different gametes. This separation is called by Mendel as segregation. This separation of factors responsible for tallness and dwarfness during the formation of gametes is called by Mendel as segregation. Okay, and this is called as the principle of segregation of unit factors, which is the third deduction. Mendel said that unit factors are in pairs. Just we have discussed it. Capital T, small t. So why they are in pairs? Because Pisum sativum is a diploid individual. A diploid individual has chromosomes which are in pairs, so that one carries gene for or factor for tallness and the other carries factor for dwarfness. Why? There are two, because one is received from father, paternal in origin, the other is received from mother, maternal in origin. So father is pure tall, so this F1 individual got capital T gene, and mother is said to be pure dwarf in parental generation, so the F1 individual got small t, and that is the reason why there are two factors. What we all know is, during the formation of gametes, a process called as meiosis takes place. What is meiosis? Meiosis is the reduction in the number of chromosomes during the formation of gametes. How the reduction takes place? One chromosome, that is paternal, goes to this is a first pair of chromosomes, let us say, consider. Out of the first pair of chromosomes, one goes to one sperm, the other goes to another sperm in human beings. Likewise, one goes to the first, in the first pair of female also, one goes to one ovum, the other goes to the other ovum. The same thing happens in Pesum sativum even. What happened here? Out of the two factors which are present on a pair of chromosomes, as one chromosome got into one gamete, the gene or factor capital T present on the same chromosome went into that gamete. Likewise, both the factors capital T and small t got separated and entered different gametes. This separation of the factors responsible for a pair of contrasting traits of a single character during the formation of gametes is called as segregation by Mendel. So, how factors segregate? Because meiosis occurs in sexually producing organisms, which reduces the chromosomal number to half, so that segregation takes place. Okay, so based on these derivations from a monohybrid cross, Mendel postulated that three principles. Mendel postulated three principles: principles of unit factors, principle of dominance, principle of segregation. So these are the three postulations derived by Mendel based on monohybrid cross. So I'll give you 30 seconds. Please write down these three. Mendel's postulates based on monohybrid cross. First one, principle of unit factors. Second one, principle of dominance. Third one, principle of segregation. After completion, tell me whether you have written it or not. Just tell me done, sir. Anyone?
Done, sir.
Okay, good. So let us discuss the principle of unit factors. So what is a factor? We have to know. Because Mendel is a mathematician, isn't it? He is a botanist, he is a statistician, he is a mathematician, he is a specialist in culture of fruits and grapes, pomiculture and viticulture, what not everything. So when you formulate or go for any mathematical principle, if you don't know what actually that particular number is, then you mention x or y, isn't it? When you are a statistician, you call this as a factor. Something that is not known but still it is there, then you call it as a factor. So Mendel knows that the plant is tall. Why the plant is tall? Because there is something inside the plant for tallness. But he doesn't know anything about that. But he knows that there is something inside. If you see red color. So why you call it as red color? Because there is something that gives it red color, isn't it? Carrot is said to be orange color. Why it is orange in color? You say there are certain carotenoids or some bilins, phycobilins are there, isn't it? But until and after the discovery of certain carotenoids or phycobilins, you don't know what actually is present. But when you see a carrot, because it is orange in color, you say that it is orange in color because something for orangeness, orange color is present inside. And that something which is not known is called as a factor. So Mendel thinks that there is something for tallness, there is a tall factor, and something for dwarfness, there is a dwarf factor in the parental pure line generations. Okay, so Mendel coined the term factor for these units of heredity during his experiment, because these factors are transmitted from one generation to the another generation. These were considered as the units of heredity. So what are Mendel's factors? Mendel's factors are units of heredity. The units which code for trait during the process of inheritance are called as factors. And what is the principle of factors, unit factors? During the process of inheritance, offspring receives one unit factor from each parent. So the F1 tall is said to be F1 tall is said to be a hybrid tall because it received capital T from the father and small t from the mother. So thus the offsprings, even we, receive one unit factor from each parent. Thus there are a pair of factors responsible for a pair of contrasting characters of a single trait, which we call this as the principle of unit factors. Okay, thus unit factors according to this principle are always in pairs because Pisum sativum is a diploid individual. So is this principle applicable to all the characters in the world, in plants and animals? No. Why? Because in haploid plants there is only one unit factor because there is only one chromosome. One chromosome carries only one factor. So the principle of unit factors according to Mendel, which says that unit factors are always in pairs, is not universally applicable because there are haploid individuals. In addition to haploid, we also have polyploid individuals, isn't it? 3n, triploid, 4n, tetraploid, and so on. For 3n, there are 3 chromosomes. That means for stem length, how many? There will be 3 factors or 3 genes. If it is a tetraploid, there will be 4 factors. So in such cases where there is haploidy and polyploidy, Mendel's principle of unit factors, which says that unit factors are always in pairs, is not universally applicable. But in diploid individuals, wherever you come across in the world, whatever the plant, whatever the animal may be, if it is a diploid, according to Mendel, the factors, unit factors, are always in pairs. So for all the diploid individuals, this principle is universally applicable. And coming to the second principle, the principle of dominance. So what is dominance? When two plants differing in a pair of contrasting traits are crossed. So there are two plants, one is tall, the other is dwarf. When two plants differing in a pair of contrasting traits, what are the contrasting traits? Tall and dwarf. What happened here? They were crossed. The trait which appears in F1 generation, in F1 generation, all are said to be tall only. The trait which appears in F1 generation is called as dominant trait, and the one which does not appear in F1 generation, being suppressed, is called as recessive trait. That is what dominance is. Then what are dominant and recessive factors? The unit factor responsible for dominant trait is called as dominant factor, and the unit factor responsible for a recessive trait, that is dwarf, is called as recessive factor. Then what is the principle of dominance? Out of a pair of factors, determining a pair of contrasting traits, one for tallness, the other for dwarfness, where in a hybrid, what is that? F1 plant. In a hybrid, one factor, that is capital T, which is dominant factor, suppresses the other factor, that is small t, which is called as the recessive factor. This is the second principle of dominance. Okay, I am giving you one example. Okay, so let's say the flower color of Pisum sativum. Flower color of Pisum sativum. One is purple, the other is white. Okay, they were crossed, and you got some result. Based on result, you have to say, based on the result between the cross between purple colored flower plant and white flower plant, you got some result. Based on the result, you have to say which one is dominant and which one is recessive. So can anyone tell me, out of purple and white, which one is dominant and which one is recessive? Purple is dominant and white is recessive. How can you say that?
Because the F1 plastic is attracted to the purple flower.
Because F1 plants are having purple flowers. Okay. What happens if F1 plants are white in color? Flowers of the F1 plants are white in color. What will you say now?
Then white is dominant.
Then white is dominant and purple is recessive. So just by seeing two different plants, we cannot say which one is dominant and which one is recessive. We have to cross the two contrasting traits, see the F1 generation and the character which appears in the F1 generation. Based on that one only have to determine whether it is dominant or recessive. Thus, you can determine the dominance and recessive relationship only in a hybrid. Only in a hybrid, that means heterozygous condition only you can say this, but not in a homozygous condition. If someone gives you a tall plant, capital T, based on the tall plant, you don't know anything about the factors. Based on just seeing the phenotype, can you say it is dominant or recessive? No. Someone gives you a dwarf plant only. Can you say it is dominant or recessive? No. Only when they are crossed, and in the next generation, if dwarf appears, dwarf will be dominant, tall will be recessive. If tall appears in the F1 generation, then tall will be dominant and dwarf will be recessive. Because Mendel got only tall character in the F1 generation, tall is said to be dominant and the dwarf is said to be recessive. Okay? Next. Coming to the type of dominance called by Mendel. Mendelian dominance is called as complete dominance. Mendelian dominance is called as complete dominance. As one trait, that is tall or purple or round shape of the seed, completely suppresses the other contrasting trait. That means tall suppresses the dwarf, purple suppresses the white, brown suppresses the wrinkle character. Without any intermixing. That means a tall plant is crossed with a dwarf plant, only tall appeared. There is no intermediate phenotype. That means size which is intermediate between tall and dwarf is not found in the F1 generation. A purple flowered plant is crossed with a white flowered plant, and he got only purple flowers, but not light purple, dark purple, intermediate purple, and semi-white, half white, full white, nothing like that. Only purple he got. That means the purple color completely suppresses the white color, and such a dominance is called as complete dominance. Please write this point. Mendelian dominance can be called as complete dominance as one trait completely suppresses the other contrasting trait in a hybrid. You can mention this one also. In a hybrid without any intermixing. If white color intermixes with purple color, what happens? Purple will be light purple. But there is no intermixing. All the F1 are said to be purple colored only. Okay? So this is what we call Mendelian dominance as. So can you say the Mendelian dominance is universally applicable or not? No. After Mendel's discovery of principles of inheritance, several scientists became geneticists. They have done a number of experiments on what we call genetics, and they have come across with some other principles called as incomplete dominance and codominance. And when there is an incomplete dominance and codominance, Mendelian dominance is not applicable. So Mendelian dominance, which is a complete dominance, is not universally applicable, because there are certain other phenomena called as incomplete dominance and codominance, which we'll discuss in the coming classes. Okay? Next. The last one is the principle of segregation. What does it say? It is the segregation or separation of unit factors. Separation of unit factors, one into one gamete, the other into another gamete, during the process of formation of gametes is called as segregation. Based on this, Correns, as I have mentioned before, It is written in certain books. Correns proposed the first law of Mendel. This first law of Mendel is called as law of segregation. Okay, and here you have to remember one thing. The F1 plants. Can anybody respond? F1 plants are pure tall plants or hybrid tall plants?
Hybrid tall plants.
Very good, excellent. So F1 plants are hybrid tall plants. So this F1 hybrid gave rise to gametes. Can anybody respond? Are the gametes hybrid or pure?
Pure, sir. Pure, sir.
Pure, because it carries either T, capital T or small t. Either dominant factor is present or recessive factor is present, but both are not present in the same gamete. Thus, the gametes are pure for a particular character. The gametes, though the F1 plant is hybrid, its gametes are pure for a particular character. And that is the reason why this is also called as the law of purity of gametes. So you can write it down. Mendel's first law, that is law of segregation, is called as the law of purity of gametes. Okay? Clear?
Yes, sir.
Okay. Now we'll move to the second law, that is the law of independent assortment. So the first three principles proposed by Mendel, that is principles of unit factors, principle of dominance, principle of segregation, which was made later into law of segregation. The first law of Mendel are said to be based on monohybrid cross, and the fourth principle, that is independent assortment, is the first postulate of Mendel, which was made into the law of independent assortment, is based on the dihybrid cross. What is dihybrid cross? It is a cross between two plants differing in two pairs of contrasting traits of two different characters. So here we take two characters, that is seed shape, seed coat, color. So seed shape is said to be round or wrinkled, seed coat color is said to be yellow or green. Round is said to be dominant, wrinkled is said to be recessive. And hence, round is represented by capital R and wrinkled by small r. So round and wrinkled. Likewise, yellow is dominant and green is recessive. So thus yellow is represented by capital Y and green by small y. And we have taken one plant whose seeds are round and yellow in color, and crossed this plant with another plant whose seeds are wrinkled and green. Why he has done this so? Mendel wanted to know why the round seeds are yellow in color, why the wrinkled seeds are green in color. Why not the round seeds green in color? Why not the wrinkled seeds yellow in color? In order to know this, what you have to do? You have to go for a hybridization experiment. So Mendel did hybridization experiment. So in his experiment, Mendel considered two pure lines, true breeding plants, differing two characters, that is seed shape for round and wrinkled, and coated in color yellow or green. Round is dominant over wrinkled and yellow is dominant over green, as we all know. And parental generation, what happened is Mendel crossed a true breeding pea plant having round and yellow seeds, this one, with true breeding plant having wrinkled and green seeds. So this one. And in the F1 generation, all the plants showed were round in shape and yellow in color. So round and yellow characters appeared in the next generation. Why? Because round suppressed wrinkle because it is dominant, yellow suppressed green because it is dominant. So only dominant characters appeared in the F1, and then we can say that this F1 individual is a hybrid. Because the parents are pure, this can be hybrid. And what happened? The F1 individuals were now self-pollinated. So round and yellow are self-pollinated. So this represents the androecium, this represents the gynoecium of the same plant because it is a bisexual plant. Capital R small r, capital Y small y. And now what happened is, offsprings of F2 generation showed all four phenotypes. That is, round and yellow as in the parents, wrinkled and green as in the parents, and two new combinations, round and green and wrinkled and yellow. And these two are called as parental combinations, and these two are called as recombinants or non-parental combinations. Okay? In 9 is to 3 is to 3 is to 1 ratio. So here, based on this cross, we can deduce the genotypic ratio. How? Mendel observed 9 genotypes. That is, the first one is capital R, capital R, capital Y, capital Y. How you have to distinguish? It is only one genotype. And number of plants with capital R, capital Y, small y, they are said to be 2 in number. Okay? So 1, 2. Next genotype is capital R, small r, capital Y, capital Y. How many are they? 1, 2, again 2. And the next one is capital R, small r, capital Y, small y. How many are they? They are 4 in number. So 1, 2, 2, 4. How many? 9. 9 genotypes are there for what we call, 9 plants were there for round and yellow character. Of this, how many are there? Only 4 genotypes are for round and yellow. So for round and yellow, Mendel got how many? 4 genotypes. Next one is capital R, small y, small y. Capital R means round, small y for green, and round and green seeds. So he could get the round and green seeds in the F2 generation. How many are they? Capital R, small y is 1. Another combination of the round and green is capital R, small y, small y. How many are they? They are 2 in number. So, for round and yellow there are 4 genotypes. For round and green there are 2 genotypes. Here there are 4 and here there are 2. And the third one is small r, capital Y. How many are there? Small r, capital Y is 1. Small r, capital Y, small y is 2. So these are wrinkled and yellow. This is the new combination that Mendel got. Like round and green. How many are they? There are 2 genotypes for wrinkled and green. And last one is small r, small y, small y. And this is only 1. Thus he got 1, 2, 3, 4. 4 phenotypes, round and yellow, round green, wrinkled yellow, wrinkled green. And for round and yellow there are 4 genotypes. For round and green there are 2 genotypes. Wrinkled and yellow 2 genotypes, and wrinkled and green 1 genotype. 4 plus 2 plus 2 plus 1, that is total there are 9 genotypes observed by Mendel. So you can simply draw like this. For round and yellow, for wrinkled and green, and this is wrinkled and yellow. This is wrinkled and green, and these 3, the remaining 3 are said to be round and green. Okay. Next. So how did Mendel explain this? So Mendel said that true breeding pea plants of parental generation, that is round and yellow and wrinkled and green, produced gametes in the following combination: capital R, capital Y, small r, small y. If these gametes upon fertilization, what happens? They produce plants with the dominant genotype called as hybrids. That is capital R small r, capital Y small y. How he got this? Okay. Next one. The F1 individuals were self-pollinated. Round and yellow F1 individuals were self-pollinated. The F1 plants during self-pollination produced gametes in 4 combinations. Capital R, capital Y, capital R, small r, capital Y, and small r, small y. This is because of the fact that what the Mendel says is factors for round and yellow, that is capital R, capital Y, that is capital R and capital Y, during gamete formation not only formed parental combination, that means capital R separated from capital Y. Okay, so only capital R is present and it combined with small y. Capital R not only formed combination with capital Y, that means for yellow color, but also formed a combination with small y for green color. Likewise, small r and small y, they formed the same combination and they got separated from each other and they formed another combination with capital Y. So in addition to the parental combinations, non-parental combinations were formed in the gametes itself. Why this happened? Because let's see in the previous slide. In the previous slide we have seen that in the gametes also capital R and capital Y are together. And in the gametes of the wrinkled green, small r and small y are together. But here, in the next generation, what happened here? Capital R got separated from capital Y because the gene for seed shape, that is round, behaved independent of the gene for Y type. These are now independent of each other. They are said to be separating from each other. That means R cannot inherit itself with capital Y only. Capital R can inherit without capital Y. Thus they are independent. So this independency gave rise to 4 different combinations, 4 different combinations, of which 2 are parental and 2 are said to be non-parental. Likewise, factors for genes for wrinkled and green also, smaller and small y, also formed parental combinations and behaved independent of each other and assorted. What is assorted? Again, separated from each other. Assorted, separate from each other and formed new combinations or recombinations. So this is called by Mendel as independent assortment. How? The genes for seed shape, capital R and small r, behave independent of the gene for seed coat color, capital Y and small y. So in parental combinations, capital R, capital Y, small y present. But as these two are behaving independent of each other, capital R instead of combining with now with small y, it also combined with small y. Capital Y, it also combined with small y. Likewise, small r instead of combining with small y, it combined with capital Y. Thus 4 combinations are produced, and this phenomenon is called by Mendel as assortment. Because these two exhibited independency to each other, it is called as the principle of independent assortment. So Mendel concluded this like this. Mendel postulated that during gamete formation, segregating pairs of unit factors, that is capital R small r, capital Y small y, assort independently of each other. They never form only the parental combinations. If only parental combinations are formed, what happens? These are the gametes that are formed in the next generation. Then what happens? When these cross with each other, capital R small r, capital Y small y. When these two cross each other, capital R small r, capital Y small y. When then all plants will be only round and yellow only. But he got 4 different types of phenotypes: round and yellow, round and green, wrinkled and yellow, wrinkled and green. Why? Because these two got separated from each other, these two got separated from each other. And such a random segregation is called as independent assortment by Mendel. Okay. So this is all about the four postulates of Mendel. So Mendel's findings went into dark. Why? Contemporaries tend to dismiss Mendel's numbers and ratios as merely empirical and devoid of respectable theoretical framework. So, and till Mendel explained the principles of inheritance, how the process of inheritance or heredity takes place, no one knows about this. So they wanted a theoretical background for this. But there was no theory established at that time. Mendel even does not propose any law. He just made his results published. Okay, and Mendel was discouraged by the lack of response from the scientific community, and the results obtained on heretium, as I have said before, also were discouraging. And hence Mendel's findings went into dark and were ignored for how many years? 3 decades, until their rediscovery. Okay. So what are the reasons for obscurity of Mendel's findings? Obscurity means not coming into light. Biologists' preoccupation with Darwin's theory of origin of species, which appeared in 1859. So Mendel published his findings in 1865, and everyone is going after the evolutionary theory proposed by, before 6 years prior to this, by Charles Darwin. So everyone is after Darwin, and Darwin is said to be such a famous scientist. And though Darwin and Wallace both proposed the natural selection theory, we attribute the theory only to Charles Darwin. Why? Because when you publish something, you go to the public. You have to attend a conference, you have to attend a seminar, you have to attend a symposium where you have to project your ideas, your publication, your results, and get them into debate. Okay. And only Darwin appeared in all the conferences, not Wallace. So we consider the natural selection theory as proposed by Darwin, which is not true. But the same is case here. No one knows about the principles of inheritance because Mendel does not propose any theory, whereas Darwin has proposed a theory and everyone is after it. So no one is bothered about Mendel's findings. Okay. Second one is obscurity of the journal. The Mendel's publications were given in a journal which is restricted to only Austria. So no one knows about it. And even Mendel is not going away from Austria to defend his publication because he failed in producing the same results with respect to heretium. That is the reasons why Mendel's findings being produced in a local journal are said to be another reason for obscurity of Mendel's findings. And third one is failure in getting same results on the hawkweed heretium and the advice of Nageli and Kramer, which proved later to be a parthenogenetically reproducing plant. Why Mendel did not get the result on heretium? Mendel is working on Pisum sativum, which is a sexually reproducing plant. So Kramer and Nageli would have given a sexually reproducing plant for Mendel, but rather they have given a plant which is parthenogenetically reproducing, a sexually reproducing plant. That means here the plants are produced from only the unfertilized eggs. So when you are producing plants from unfertilized eggs, how can you do this? How can you get the same results like the one that got a sexually reproducing plant? So that is the reason, third reason for Mendel's findings obscurity. And in 1900, 3 different scientists, De Vries, Correns, and Schermark, rediscovered Mendel's laws, and they have published and they have given full credit to Mendel, and they have published it in the form of laws, law of segregation and law of independent assortment. So Hugo de Vries is a Holland scientist. He worked on 15 different varieties, and he published his paper on the law of segregation of hybrids. That is something similar to Mendel's findings. Karl Cornens, he is working in Germany. Okay, sorry, this one here. He worked in Germany, also found Mendel's publication and published it as Gregor Mendel's Law. And in 1902, after the rediscovery, Bateson published, but by translating the entire work of Mendel from Austrian to English, he published it as Mendel's Principles of Heredity. And at the same time, Sharma from the Austria have conducted similar work to Mendel and discovered his paper while searching references. One has to refer. I have searched for 70 years whether the work that I am going to take up for my PhD is done by any other or not. But these have not done this. But while Sharma is searching for references, he could find Mendel's publication, and this was brought into light by these three great scientists. We have to call them as great scientists. Why? Because they can no one knows about Mendel except these three. They can put it aside and give the full credit to themselves, but they did not do so. So as Mendel is great, for me these three scientists are also great scientists. Okay, so Mendel's laws were proposed by these three rediscoverers, especially Correns, and the first law is said to be law of segregation, called as first law of Mendel. It says that when two factors responsible for a pair of contrasting characters of a character are brought together in F1 hybrid, they do not contaminate or mask each other. That means capital T, small t. They will not mix up with each other, but rather they separate during the formation of gametes, that is at the time of gametogenesis, so that each gamete receives only one allele, either T or small t, of each trait. Hence, this process is called as segregation. This separation of gametes during the formation of separation of factors during the formation of gametes in a hybrid is called as law of segregation. It is also called as law of purity of gametes. And why segregation is significant? It refers to Mendel's proof that recessive traits reappear in predictable patterns. So, though in the parent generation dwarf is present, in the F1 generation only tall is present, dwarf did not appear. Because segregation is there, dwarf reappeared in the F2 generation in 3:1 ratio. So, segregation is significant that recessive traits reappear in predictable patterns. In later century, molecular studies revealed that chromosomes do separate during gamete formation. So, genes, that is Mendel's factors, present on chromosomes also separate. This is what we call it as Sutton's chromosome theory of heredity, which we will discuss while we are discussing linkage theory. Okay? Concept of linkage. And coming to the law of independent assortment. This is also called as the second law of Mendel. What does it say? It states that when the parents differ from each other in two or more contrasting characters, for example, round and yellow, wrinkled and green, seed shape and seed color, then the inheritance of one pair of factors is independent of, that means capital R small r and capital Y small y. Inheritance of one pair of factors is independent of the other pair of factors. And what happens here? They separate from each other and they form new combinations in addition to the parental combinations. Hence, this is called as the law of independent assortment. So, coming to the critical review of Mendel's principles. So, with this, we conclude the Mendel's laws. And tomorrow, we are going to discuss about the critical review of Mendel's laws. Whether four principles of Mendel, that is, principle of unit factors, second one, principle of dominance, third one, principle of segregation, fourth one, principle of independent assortment. In the next class, tomorrow's class, we are going to critically review all these four principles, whether they are universally applicable or not. Based on that, we are going to based on after tomorrow's class, we are going to conclude Mendel's laws. Okay. So tomorrow, what we are going to see is principle of unit factors a universal phenomenon or not? Is principle of dominance a universal phenomenon or not? Is law of segregation a universal phenomenon or not? And is independent assortment universally applicable or not? If so, why? So all these things we are going to discuss tomorrow in tomorrow's class. So don't miss the class. Because it is not generally discussed in your classes. Okay, for competitive examinations, you need to dig deep into the subject. So don't miss tomorrow's class. So with this, I conclude Mendel's laws. If any doubts you have, please ask me. Otherwise, we'll close the class now. Thank you.